It's full screen. Okay, it's full
Yes.
screen now. Fine, thank you.
So our topic of discussion today is comparative estimation of catalase and peroxidase from taproot and hairy root of carrot. Now, before moving further, let me tell you this catalase and peroxidase are the two important enzymes required for the basic metabolic activities in the plant system. Now, why we need to estimate these two important enzymes? Now, you see, the last two classes we have discussed about the plant tissue culture techniques and applications of it. Now, in many cases, after tissue culture, the plants develop some extra vigor or some diminished behavior in some cases. In some cases, plants behave differently from their wild type variants. So to determine how they are affected physiologically or whether the tissue culture plants are responding well physiologically, we need to calculate the enzyme activity. And these two enzymes are a very well representative of the whole enzyme class or the whole physiologically active enzyme class of the plant system. So for this class, we have chosen these catalase and peroxidase enzymes. Now, every living species, not only plant, humans, all other animals, everything, bacteria, virus, except virus, bacteria, fungi, everywhere, there is a particular chemical group called reactive oxygen species, or commonly known as ROS. Now, these ROS are actually the molecules containing an free oxygen atom, or you can say oxygen atom with an unpaired electron in its outer shell. Why they are very important? These molecules are very important because they are extremely reactive. You know why the bleaching powder is too much reactive? Because after reacting with water, bleaching powder releases chlorine in its nascent stage, that we called as nascent chlorine, because it has a free unpaired electron in its outer shell. That's why it is extremely reactive. Reactive oxygen species is exactly like that. It will react with almost anything, and it has a huge capacity of penetration. Like if reactive oxygen species amount is too much, they can, as I tried to explain in this image, if reactive oxygen species, if the number of reactive oxygen species started to getting higher, they can easily break the cell membrane. They can also break the nuclear membrane. They can also break other membrane organelles like mitochondria, chloroplast, everywhere. So actually, these reactive oxygen species lead to a higher excessive damage of the cell if their concentration is not checked. Now, these reactive oxygen species or ROS are of two types. In one cases, as you can see, follow my pointer, that there is a superoxide radical O2 minus here, and another is the hydroxyl radical. Hydroxyl radical means OH minus, OH with a free unpaired electron. And do not mix it with hydroxyl ion. It is different from hydroxyl ion. The reactivity of hydroxyl radical is much higher than that of hydroxyl ion because, as I mentioned, the electrons are unpaired. So the unpaired electrons, what they try? They always try to get paired and to form a stable bond. And during the bond formation, they do the reaction, and in most of the cases, as a side effect, cellular membrane damage occurs. And there are lots of membrane-bound organelles like mitochondria, chloroplast. If in a plant cell, this reactive oxygen species started to get higher, then these membrane-bound organelles, the mitochondria, chloroplast, will started to get disintegrate. And if what will happen if mitochondria get disintegrate, the cell can no longer do the respiration. In plants, the chloroplast membrane starts to disintegrate. Then what will happen? The cells cannot do the photosynthesis. So ultimately, it will lead to the plant death. So these two enzymes, as I mentioned before, peroxidase and catalase, these two enzymes are the very important enzymes in the production of this reactive oxygen species. One enzyme produces hydroxyl radical, and another enzyme produces superoxide radical. So in terms of measuring this enzyme activity, we can deduce the amount of reactive oxygen species within the cell. Say if the catalase or peroxidase activity is much higher than normal, then we can easily conclude that, okay, since these enzymes are very active at this moment, that means they are producing too much superoxide and hydroxyl radicals inside the cellular system. So this is our target. This is the rationale behind this study, why we are going to this study these two enzymes. So any types of enzymatic, most of the, not any types, most of the enzyme study required a specific machine called spectrophotometer, and the process is called spectrophotometry. Now, this spectrophotometer, this is a very sophisticated machine, but there are other versions also available. So instead, I tried to explain the basic structure of this machine in a simplified way. So in each and every solution has its own optical density. Whatever solution it may be water, it may be alcohol, it may be some bacterial solution, it has its own optical density. Now, what is optical density? When you will pass light through a solution, suppose you put a glass of water in a uh, uh, glass of water, and you put a specific high power torch in a dark room, you put a high power torch in front of the glass. The whole light will not be passed through the glass. Some light will be reflected, some light will be refracted, and some light will get absorbed by the water. And only after absorbing, some light will get refracted and will reach the other end. This absorbance of the light by the solution or the water or whatever the solution is within the glass is the optical density of that solution. The term optical density also, you can, for simplicity at this moment I am using absorbance, is the very common in any types of chemical reaction. This term you will come through, the optical density or absorbance of a particular solvent or solution. Now what is happening in this machine? This is the light source, or as for simplicity what I told, that to put a torch. So this is a light source, and this is an entrance slit. This will make sure that the light source moves towards the right path, and also this will make sure a particular single dose or single beam of the light will enter. So the light source, and through the entrance slit a single beam will enter. Where it will enter? It will go and bounce to a prism. Now what prism do? It will refract the light and produce a VIBGYOR. So that's exactly what happened. It attacked the prism, and during refraction, when it emitted from the prism, it produced a VIBGYOR. And at the end of the VIBGYOR, there is the exit slit. Again, exactly like the entrance slit, the exit slit function is to allow only one particular beam to exit. So the VIBGYOR, all the 7 beams, all the 7 colors cannot exit all together. The exit slit will allow only one color to exit. So in this image, you can see that through the exit slit, only the green can exit from the slit. If you change the exit, in very old models, there usually a rotor by which you can change the exit slit or change the wavelength, or in this sophisticated machine, everything is controlled by software. In the software, you can put a specific wavelength, and say you put a specific wavelength of red, and the exit slit will move towards the red. So only red will pass through them. So this way it will manage the pass of the single beam of light, which will ultimately is your target container, or it's at this moment I am suggesting it as a container. I am going to explain this particular structure more details in the next slide. At this moment, consider this is the glass with a water, and it will ultimately attack with your glass of water, and it will pass through it. Some will get absorbed in the system, and ultimately it will get detected in the detector, and you will get the optical density on the screen of the machine. This is as simple as it is in a simplified way. This I think this at this moment enough. I'm going to the more. I'm going to more detailed discussion in this upcoming slide. So this is a basic principle of the spectrophotometer. Now this spectrophotometer works on a mathematical principle that is known as Lambert-Beer law. Now before going to this exactly Lambert-Beer law, I told you that I'm going to discuss more about this glass in the next slide. So this is actually our glass in scientific term. In scientific terminology, we are going to name this as a cuvette. This normally being made up of with quartz, and the unique thing about this is the diameter, the width of the cuvette is actually constant. So in most of the cases, it's 1 centimeter, but it is constant. So by taking the cuvette or putting the solution in it, you can easily say that I have put this much amount of solution in it. So this is the purpose of the main, that it will give you a specific volume of the solution. So you'll get a specific volume that, okay, this is the amount of my solution, and also you'll get exact the area which the light is traversing. This is required when you are going to measure the optical density. So this is your incident light, and this is your solution within the cuvette. Now everything is known. Since you know the length and the height of the cuvette, you can get the volume of the cuvette and as well as the volume of the solution. And this is your transmitted light. Now, two mathematicians, August Beer and Lambert, they developed a mathematical equation by which the absorbance of a solution or solvent or a material can be determined using the optical density. Now, this is the absorbance of the solvent, in this case, this, and this is the I1. I1 is the transmitted light. Normally, in some cases, we put I1 or I1, this one is the transmitted light. You can see the lower intense beam, and I0 is the incident light. You can see we have put it in a darker color because at the time of incident, normally every light is in a higher, it's much brighter, and after absorbance, the brightness will reduce in a simplified way, in layman's term. And I think now it's clear that incident light, I0, transmitted light, I1, and we are going to take the negative logarithm of I1 by I0. So this will give you the epsilon into CL. And what is epsilon? Epsilon is called the extinction coefficient. Someone called extinction coefficient, some called molar extinction coefficient. Both of them are same. Epsilon is the extinction coefficient. It is a type of constant, and it is available. It varies from sample to sample. But for a particular sample, say for water, the epsilon will remain constant in everywhere. Say for milk, the epsilon will remain constant everywhere. It may vary from sample to sample, but in a particular sample, under any circumstance, it will remain same value. That's why it was given the name constant. Now, C is the concentration of the solvent or the solution, and L is the path length. This is why path length is very important. Now, this path length is required while getting the calculation. And now using this particular cuvette, you know the path length, because the cuvette are of specific breadth or length and everything, width. So this path length is long, depends on the cuvette, but it is known to the scientist. So based on this, actually our target is to get the concentration. Now in the machine will give you the reading, which is that in the particular sample, you will know the epsilon, it is available in literature. You know the length, because I told you the cuvette length is constant. In each and every cuvette, there is written what is the length of it. So it is constant. So you can get the value of C, or the concentration of the sample. So this is a way, by using spectrophotometry, you can determine the concentration of a solution. So another machine will require, apart from most of the 90% of our work will be taken by the spectrophotometer machine, but another small amount of work will be done in the machine called centrifugation or centrifugation machine, or the process also called centrifugation or centrifuge. Lots of name are there. In some cases you will find ultracentrifuge, in some cases find analytical centrifuge, but the centrifuge and centrifugation theory is everywhere similar, whatever the name be. Now this centrifugation actually comes from a very known two forces which you know already, that one is centrifugal force, another is the centripetal force. As if you can recap your class 2 physics, that when you are moving a ball in your hand, the force acting outside is the centrifugal force. There is a force which wants to take the ball outside or out of your range, and another force is acting on the ball towards your hand, which wants to take the ball towards you, is called centripetal force. And the fine balance of the centripetal force and the centrifugal force actually keeps the ball in the exact axis. This is the same in every cases. So based on this very simple theory, actually it has been found there, scientists have designed a very wonderful machine. This is the centrifugation, and you can look that this is called rotor, and there are small holes in it. Now when you prepare your sample in these small tubes, you put your sample. Now why we require this? Say your sample may contain your, say you prepare in a simple way, you prepared a solution of sugar and water. Now we put too much sugar in it, so the sugar is cannot getting dissolved because you have put too much sugar in it. Now how you will get the sugar out, or how you will get the water out without the sugar? It is, in normal sense, it is very difficult, but you want that the sugar, I need the sugar out. Yeah. So in one way, we see the sugar particle has a different density and the water has a different density. So when they will be moved, when they will be rotated in a high speed, the sugar particle will get will get pellet down, or we use the term pellet down, in the lower part of the tube, and the water will remain on the upper part of the tube. This is actually very required in enzyme assay because we are going to use plant samples. Plant samples are actually solid samples. We are going to crush them in some solvent, and we are going to extract the inner material in the solvent. But we don't need the crushed plant sample. We need to move them out. So how to differentiate between the solvent and your plant sample? This is the way you can do it. So this is the exactly same principle. We put your tubes, the solution tubes, and start the machine. It will move in a, it will rotate in a very high force, and exactly same principle. And after some time, the higher density solutes will start to settle down, and the lower density will become on the upper side. And we just take the upper side by using a micropipette or a pass-through pipette, depends on the size. So this is in a simple way. Now this rotor can be of two types. One is fixed angle rotor. In most of the cases, in most of the laboratory, we use fixed angle rotor. Even in this particular experiment, whatever rotor has been used are fixed angle rotor. But there are other types of rotor called swing bucket rotor. These are very costly, of course, swing bucket rotor, because from the diagram you can see that you can actually manipulate this attachment side so that you can change the angular angle of the attachment. And based on this angle of attachment, actually, you can change the relative centrifugal force, or RCF. And it actually depends on if your sample contains 2 or 3 types of different solute, then 2 or 3 types of different solute you can get separated from this swing bucket rotor. But for simplicity at this moment, we'll attach with the fixed angle rotor where there is a fixed angle and will be useful to get separate a single type of solute from solvent. Now this equation is very important, RCF, relative centrifugal force. Now one thing is important, why I'm saying relative? Because this relative terms comes this centrifugal force. Now centrifugal force actually depends based on the diameter of the rotor. If the diameter becomes small, the centrifugal force changes. If the diameter becomes large, the centrifugal force changes. And this R in R suffix centimeter is actually the diameter of the rotor. So this is why the term came relative. So whatever value is coming is relative to that particular machine only. That's why the term relative. Okay. So this is the basic of centrifugation. This now we are moving to the experiment part. So just a minute. Yes, thank you. Now we are moving to the experiment part. So there are some prerequisite for this experiment. Any types of enzyme assay, why it's not any types of enzyme assay, there are some prerequisite. What are they? One is crushed ice. It's extremely important. Another is chilled mortar and pestle. Another is polyvinyl pyrrolidone, or you can say PVP or PVPP. All actually all of these are same, polyvinyl pyrrolidone or PVP. These three things, one is the chemical PVP, another is chilled mortar pestle, another is crushed ice. They are very important. Let me explain why. Enzymes, when they are being extracted, except when they are not in the, they are within their cellular environment, when they are outside their cellular environment, are very susceptible to heat. They are extremely susceptible to heat. Seeing little bit of heat, they are going to degrade. And this is a schematic of it. Little bit of heat, little bit of change in magnetism, little bit of change in pH, the enzyme is going to degrade. This is a normal proper enzyme. This is the denatured enzyme. And once you will get degraded, once the enzyme will get degraded, the enzyme assay will give you a negative result. So that will not tell you whether your sample contains that enzyme or not. It will give you a blank result. So you need to be extremely careful when you are dealing with the enzymes. This is why we need crushed ice and the chilled mortar and pestle. The mortar and pestle normally before extraction of enzyme has been kept in -20 for a period of 12 hours. In -20 degrees Celsius for a period of 12 hours. They will become extremely cold. In that cold mortar and pestle, we will crush the plant sample and make it powder using liquid nitrogen so that the water from the sample will get out and it will become easy to crush, and we'll put them in a solution. I'm going to explain that. Now PVP. PVP is required because in most of the plant samples, in most of the all plants contain a group of compound called phenolic compound. Here is I have given an image of catechin. Catechin is a type of phenolic compound only. Now these phenolics, they what do? They actually inhibit the enzyme activity by binding with it. But your sample contains enzyme, but after crushing, the phenolics leach out from the sample and they bind with the enzyme. So when you are actually measuring the enzyme, you are getting no free enzyme in your sample. So by adding PVP, they will actually keep catechin busy. Catechin will bind with PVP instead of enzyme, and your enzyme will become free in the solution so that they can be assayed using our traditional method. Now I told you that after crushing, so this part, chilled mortar and pestle, put your plant sample, in our case, the hairy root, carrot hairy root and the carrot taproot. The hairy root is from tissue culture grown and taproot from outside material. You crush the sample using liquid nitrogen, add a pinch of PVP to inactivate the phenolics, and put in potassium phosphate buffer. Now another question: why potassium phosphate buffer? Why not water? Because in our, what is the function of buffer? Buffer is made up of a fixed pH. You can see it's written pH 7.2 at 25 degrees Celsius. Now, what is the function of buffer? Buffer is a fixed pH, and a buffer actually resists its change of pH. This is a basic function of a buffer. Now, when we are putting in a buffer like pH 7.2, now the cellular pH is always ranges from 7.2 to 7.4. So our target is to keep the environment or to keep the enzyme in a cellular environment as we can. Now potassium phosphate buffer of pH 7.2 at room temperature of 25 degree will ensure that the enzyme will not degrade. Because if we put it in distilled water or any types of water, the enzymes will degrade. Because the pH of the water, if pH of distilled water is 6.6, which is lower than the cellular pH. So that will degrade the enzyme. Or if not distilled water, even if you get a water of pH 7, that will also degrade enzyme because that may contain impurities. That's why we have used potassium phosphate buffer of pH 7.2 at 25 degrees Celsius. We are keeping everything as normal as we can so that the enzymes will not degrade. Now, in this case, we have used a refrigerated centrifuge or cold centrifuge. Why? Again, the same reason. Again, the same reason. The centrifuge I have shown, okay, this one is also refrigerated. There are another kind of centrifuge called tabletop centrifuge. Those are not temperature monitored. But this is a very costly centrifuge because why? Only this reason we need to keep the enzymes intact. The centrifuge temperature will be set at 4 degrees Celsius. You know, 4 degrees Celsius is actually a refrigeration temperature. Our normal fridge are actually kept at 4 degrees Celsius. At these temperatures, the enzymes will become dormant, but they will not degrade. They will not be active, they will not degrade, they will become dormant. We need them exactly in that way. So after centrifugation, you see the plant material from where we have collected the enzyme actually settle down, and we term them as pellet. And in the upper region, we call this as supernatant. The supernatant is actually your crude enzyme extract. Whatever enzyme present in this plant material are now in this supernatant. And we are going to take this supernatant out and put it in another tube, and we are going to use it for our assay. This is our sample preparation. Now the assay. I am not going into the details chemistry. There is no reason to go into that much chemistry at this moment. So in a simple way, what we are going to see these enzymes, all these enzymes are actually protein in nature. So before assaying the particular enzymes, we need to check the protein content of this extract. If we see the protein content is okay, then we can say that the enzymes are present in your extract. Because enzymes, if enzymes are not present, then the protein content will also less. Now, presence of high amount protein does not guarantee that you will get this particular two enzymes. There are lots of other enzymes. They will also get extracted. Now, they will also give the protein content high. Maybe you will get these two enzymes. There is no absence of this peroxidase and catalase, but there are presence of other enzymes. They are giving you a very high protein content. But when you particularly do the reaction of peroxidase catalase, you will find a negative reaction. This may happen. Still, it is always advisable to check the protein content before doing the enzyme extract. Why? Another reason is that the buffer we have used, this is also known as the extraction buffer. The buffer in which your enzyme is being extracted. That's why it's called extraction buffer. Now this extraction buffer varies from sample to sample, from particular enzyme to enzyme. Now we need to check the protein content to make sure that your extraction buffer is at the working condition, that your plant sample is actually compatible with the potassium phosphate buffer of pH 7.2 at 25 degrees Celsius. It may happen that it is not being extracted in the potassium phosphate buffer, but it's being easily extracted of the sodium phosphate buffer of pH 7.4. It may happen. So that's why the buffer is very important, and to get that the extraction buffer is working properly, the best way to check the protein content of the crude extract. And how to do that? There is a reaction called Bradford assay using a particular chemical called Comasi Brilliant Blue. Why this name? Because after reaction, the reagent becomes a very dark blue in color. That's why it's called brilliant blue, and the reagent name is actually Comasi. After reaction, it will become blue in color. That's why it's called Comasi Brilliant Blue. That's all. In normal way, this chemical is actually normally available in two forms. One is the red form, another is the greenish form, greenish brownish form, and depends on the pH of the chemical. But in normal case, it will never be available in the blue form. It will become blue only when it will react with a protein. So this reaction is also being monitored by, I told you, the spectrophotometry. Spectrophotometer is a very important tool for us now. Now everything will be monitored by the spectrophotometer. The sample, this crude extract, will be mixed with the Comasi Brilliant Blue, and the reaction will be monitored using a spectrophotometer. And once after a 5-minute incubation period, the reaction will turn blue, and we'll monitor the absorbance in the spectrophotometer, and we'll note down the absorbance, and we can get the protein concentration. The monitoring will be done at 595 nanometer. So this is the basic protocol. Yeah, based on if we get a good amount of protein in microgram per microliter, we'll proceed towards the catalyst peroxidase assay. Now, catalyst peroxidase assay, I told you that catalyst actually converts hydrogen peroxide to water and oxygen, and peroxidase converts the hydrogen reaction between peroxidase reaction helps in the reaction between hydrogen peroxide and glycol. Now, this hydrogen peroxide production, this hydrogen peroxide breakage is very important in the plant system. Hydrogen peroxide, sorry, hydrogen peroxide, when after using catalyst, will give rise to oxygen. In many cases, this oxygen will be formed as I told before, superoxide radical. And in many cases, instead of the water formation, this H2O2 will become a hydroxyl radical. Instead of water, this will produce a hydroxyl radical. So both these reactions are extremely harmful for the plant. But those are also life-saving reaction. But the excessive amount of superoxide radical formation or hydroxyl radical formation is not good for plant. So what we are going to do, we'll take the reaction mixture, and we'll take the reaction mixture, or that is our target compound, hydrogen peroxide, and 50 microliter of the crude enzyme extract that we have extracted earlier. And then for 5 minutes, we'll monitor the OD. After 5 minutes, this absorbance will come down. Why? Because the hydrogen peroxide in the reaction mixture will get degraded, and only water and oxygen will be produced. Now, during production, sometimes superoxide radical will produce, but they will readily convert it to water and oxygen again. Now oxygen will not stay in the water, it will go in the upper part of the tube in a gaseous form. So the water absorbance of water is actually less than absorbance of hydrogen peroxide. That's why the absorbance is coming down. So if after 5 minutes we see that the absorbance of the meter is showing less reading, then you can say yes, there is presence of catalyst. Now you will normally what we do, we note down the initial absorbance and we note down the final absorbance after the 5 minutes. Similar reaction mechanism in peroxidase except in one point. Here, after 5 minutes, we are going to monitor it again like 5 minutes. We are going, except the wavelength will differ. Here we are going to monitor it in the wavelength 240 nanometer. Here we are going to monitor 436 nanometer. But the dark incubation, 5 minutes, everything is similar. But one point it will differ, that after 5 minutes, the absorbance will get higher. Why? Because the combined molecular weight of glycol and hydrogen peroxide is less than tetraglycol. The solution more turbid or more colored. So that will actually give rise to higher absorbance. So the absorbance will start to get higher. And also there will be water. So in this reaction, after 5 minutes, if we see that the absorbance is getting higher and we are going to take down the initial and the final OD after 5 minutes and see if at the end of the 5 minutes the OD is getting higher, then we can easily say that okay, that in our sample there is the presence of peroxidase. Now the reaction mixture, what will be the reaction mixture for peroxidase? We will need phosphate buffer, because we need to keep the enzyme in the cellular environment. We need glycol. Glycol is the major ingredient for this reaction, or the major target molecule for this reaction. We'll need H2O2 solution because peroxidase will not act without H2O2. So glycol and H2O2 will be reacted in presence of peroxidase. So H2O2 is also type of reagent here in this case. And we'll need the crude enzyme. Crude enzyme is very important because that will supply the peroxidase. And ultimately, after 5 minutes, it will produce the glycol. So, and in every cases, uh, uh, here the temperature I forgot to mention, but in every cases, one thing you get in your concept, that all the reactions has been conducted at room temperature, that is for 25 degrees Celsius. In 25 degrees Celsius is the optimum temperature for these enzymes. So this is the way to do the peroxidase and catalase reaction, to check the monitor, to monitor them in real time. And I told you normally we take down the initial OD and after 5 minutes the final OD, and we get difference in absorbance, and based on the difference in absorbance and the test volume, the amount of crude extract we have used, the enzyme volume, dilution factor, whether you have diluted it or not, we can actually calculate the volume activity. And volume activity not always very important, but what important is a very specific activity. So specific activity normally determined by the activity of enzyme and the milligram of the total protein. So specific activity cannot be determined without the volume activity. So volume activity will give you the basic volumetric activity of the enzyme, and from that you will get the specific activity. Specific activity is very important. Specific activity is going to tell you how much enzyme is actually present. So in a particular unit of protein, in particular unit of volume. So this is very important, and this will also give you the whether your plant sample or whether your target sample is under stress or not, because under stress the reactive oxygen species production becomes higher. So based on this, reactive with the if you found that peroxidase catalase activity is much higher in your samples, then based on that you can deduce that okay, in my sample then reactive oxygen species production is much higher. That may, from that you can infer that maybe the plant sample is under stress. So this is all in basic. I think this will be okay. So now if you have any question, you can ask. No questions? Okay, if anyone has having any problem with the microphone, they can ask question in the chat box. I'm monitoring it. Protein part. Okay, okay. Ankit, the protein part is actually which part? Most probably you are asking that why we are checking the protein content, right? Just say yes or no. Most probably that part you were asking, na? Okay. See, most of the enzymes are actually protein in nature. They are proteinaceous in nature. And besides, the extraction buffer we are using, na, potassium phosphate buffer, this is also called extraction buffer because our sample, the protein of our sample is being extracted in that buffer. Now this buffer varies from sample to sample. In all plant materials will not compatible with this particular buffer. There are also other extraction buffers. So we need to check first whether the enzyme is actually extracted in this particular buffer or not. Now, as I told you, the enzymes are actually what? They are protein. Now, if we can check that, okay, there is proteins coming in our sample, that means our extraction buffer is working, then passively we can say, okay, that means the enzymes are there in my sample. Is it clear, Ankit? It's sort of passive identification of the enzymes. Okay, my sample contains enzymes since enzymes are protein. Okay, thank you. Why were the enzymes required to be chilled? Enzymes are required to be chilled. I told you that time that enzymes are very viable to get degraded in high temperature. When they will get extracted, they has to be chilled. Otherwise, they will get degraded. Now, if your enzymes got degraded, which enzymes will get? You will get at the time of assay. At the time of assay, you will get negative result. But it may happen that actually you have extracted enzymes fine. Then, yeah, due to improper handling, they got degraded. So that's why the enzymes need to be the everything has to be chilled so that the enzymes will be in their nascent stage as can as it be. Okay. Okay. So, sir, I think That's it. They don't have any other questions.
Yeah, fine. So the a condensed form of slides we can upload in the reading material, like Pritam also uploaded,
Okay,
so
sir.
that they can use for their study.
Okay, sir. Uh, the reaction mechanism and those things are not at all uh, required. I also skip those parts.
Yeah,
So the basic parts I have discussed today, I share with you.
you make a PDF and then that can be
Yes, that can be
uh,
shared.
put it
Okay,
in
sir.
the class material so that they can study. And
Yes.
in the test when we will do, we'll ask conceptual questions. So
Yes. Yes.
see how much they understand. Yeah. Okay. If there is no other question, then we can stop the class
Okay,
here.
we can stop the class. There is no other questions.